to Chicago when I was leaving from the United States to here, there's about a 90-degree change in temperature in Fahrenheit. So this is what I came from, and I'm here to enjoy the weather and the warmth here of Vietnam. So thank you very much. At UCLA, there are probably about 10 laboratories that work on the development and applications of proteomics for addressing various questions in biology. There are many areas of proteomics that can be covered by the technology that's available. All of these are available at UCLA. Some people focus on some aspects more than other aspects. There's also a growing group of people looking at metabolites that are present in complex systems, along with developing new bioinformatics tools. But before I talk about what proteomics, what applications we do, I should maybe define what proteomics is. The proteome, of course, is the protein complement of the genome. Just knowing the genome for a particular system gives you some information on what potential proteins are present in the system. Proteomics as a field is still relatively new. The term itself was only coined in 1995. Proteomics then is the large scale of proteins in a very complex system, and how we measure them. There are various ways of how one can measure all of these proteins, but it's not so easy, as I'll tell you a little bit about today. And we focus on not only just characterizing the gene products, but also trying to identify modifications that are present on proteins, trying to measure protein interactions, trying to measure the amount of proteins that are present in a particular system. How one measures all of that, I'm going to try to give you a little bit of idea on how we do it in our laboratory. Right, so it's one thing to study a single protein, and so if you were to say this shell was to represent a single protein, many laboratories do study everything that you want to know about the single protein. But in a proteomics laboratory, one tries to study all of these proteins at the same time. We have a large collection of proteins in a complex system. And we not only try to identify what all the individual shells are in this particular reach setting, but we try to understand then how many proteins or shells are pink. We try to catalog and quantify then how many of these particular proteins are present in the system. That might be one question that we're interested in. Another question might be, are any of these proteins or shells phosphorylated? What kind of modifications are present? What is the sequence of these proteins? And we tell all of this information from the various types of ways that we measure these systems. Well, if you were to look at the genome, you could say that if you sequence the entire genome of a particular system, then you should know what every protein is present in that system. And so why you bother to look at proteomics, for example? Well, we know that because there's lots of ways that one can take the genome and translate that into proteins, that things can change during the translational scheme. There could be ways to splice proteins, to splice the RNA in order to form mature proteins. But also, proteins are post-translationally modified, and knowing the genome does not tell you whether a particular protein is modified or where the modifications could occur. And so, when one looks at, say, the human system, if the genome is coded for 30,000 genes, and you consider all the individual post-translational modifications that could be present, the number of actual protein products, or now we call them proteoforms, could be really large. I mean, this is probably just too small a number. It's probably several million different individual proteins that one could envision people being interested in measuring. So proteomics then doesn't really begin to cover all of the proteins that are present in the system, but we try to cover as much as we can and try to deliver that information in a way that's meaningful to address questions in biology. So, measuring the proteome then, there's not one single way that one could measure the proteome. Our laboratory takes a system that tries to separate the mixtures in a complex, separate them on some sort of format. I've shown a gel electrophoresis here, but we can use other ways to separate proteins. We measure proteins, the presence of the proteins, usually by mass spectrometry that Fu had discussed a little bit about in his talk this morning. And then from this mass spectrometry measurement, then we can get identification of the protein, possible modifications on the protein sequence, and try to infer some sort of level of protein abundance in that system. So with my talk then, I'm going to give you some examples then of how we do many of these types of measurements in our laboratory. I'm going to focus mostly on post-translational modifications, and also an example on how we quantify proteins, and perhaps translate some of these measurements to biomarkers that's more clinically relevant than someone had described earlier this morning. And then try to address questions in biology and medicine. So when I first arrived at UCLA in 2001, we started a project with a group led by Dr. Andre Nell in the School of Medicine. His laboratory was researching how particulates and air pollution could affect human health. This is not a current picture of Los Angeles. This is much better today when we come to LA. But air pollution is certainly a problem in many cities and many countries around the world. And we had developed a series of studies where we were trying to understand the fundamental properties of air pollutants and how that could affect human health. So we were studying diesel exhaust particles. These are these small fine particles produced by diesel engines, and how these particles then, if someone were to breathe in these particles, then how could that affect human health? What we had found was that these diesel particles, especially the very small ones, 2.5 microns or smaller in diameter, were the ones that were the most important in inducing oxidative stress and inflammation in the lungs and airway paths in the human body that could affect, that could lead to eventually death or other diseases such as asthma or bronchitis. What we had found in a series of studies that if one were to take tissues from animals that were exposed to diesel particles, or take lavage fluid from the airways, or nasal lavage fluids, either in animals or even humans, human patients, we could find proteins that were increased in people that were breathing in a lot of these air pollutants. And these proteins, some of these are chitinase-like proteins, other proteins. These were markers of people that had severe inflammation in the lung pathways. And so we had hoped then to translate some of these measurements into a way that one could decide whether a person was maybe having severe problems from the air pollution they were breathing, just by the measurement of the proteins in their nasal lavage or lung lavage fluids. This was all done then by just taking proteins from your clinical samples or from a human study and just comparing the profiles. What proteins are present in your control subject versus the proteins that were present in clinically challenged samples. So just measuring just cataloging the proteins in these two systems will give you some important information about what proteins are involved and what pathways are involved in oxidative stress and inflammation. Another study is where I'll focus on quantitative proteomics. This is a study where we're working with a researcher at UCLA, Professor Sophia Merchant, where she is interested in this single-cell organism that's photosynthetic. It's called Chlamydomonas reinhardti. It's a system that it's a eukaryotic system that grows in the dark, so it's photosynthetic. And so she's very interested in understanding what affects the photosynthetic pathways in this single-cell organism. She was involved in the genome sequencing back in 2007 of this particular species. It's also of interest because some people who want to understand or try to develop new biofuel sources, the Chlamydomonas system produces lipids under certain conditions, and so trying to harvest this as a way as a fuel source might be of interest to some. So Professor Merchant is studying this photosynthetic organism and trying to understand what transition metals has on the effect of proteins and its growth and photosynthetic properties on its system. These are just some pictures this is not Plematomonas, this is Arabidopsis, but you can see that if you grow Arabidopsis in different kinds of metal sources, you see that the colors change, the phenotype changes. But for Plematomonas, these are just flasks of the various versions of Plematomonas grown under copper or iron concentrations. You can see that the colors don't really change. So the colors by themselves don't tell you much about what the effects of metals are doing on the system. So we started a proteomics study with her several years ago where we tried to measure all of the proteins present in Plematomonas and tried to quantify them on a microgram or nanogram basis. And just try to see what proteins are increased or decreased as a result of changing the metal concentrations that the systems are grown on. And so we were able to quantify about 2,000 proteins in Plematomonas under different metal conditions, metal concentration species such as copper, iron, zinc, manganese. And from these kinds of studies, what we learned was that just simply changing the concentration of, for example, iron will change the protein complement in its proteome very greatly. This is a plot of the proteins that we were able to quantify under different iron conditions. And in this particular case, we saw maybe 100 proteins increased as a result of limiting the amount of iron in the system, and 100 proteins were decreased as a result of limiting the iron concentration. We did this for all the other metals that we were able to see. And if you were interested in the biology of then what these changes really means, you'd have to really resort to looking at these papers that we had published afterwards. But the result of the study was that there's a lot of interesting biology that one can do just by growing your species in different metal concentrations, having a laboratory that's able to measure and quantify all of these proteins at the same time, and to uncover some unusual biology. We also had a chance to measure transcript levels, or mRNA levels, as a way to compare genomics versus proteomics. And what we found was that the changes that were found at the transcript level were largely found in proteome level as well. So there was relative agreement between proteome and transcript for the changes, for the transcript changes that were found. But there were many more changes that we found in the proteome level that were not recapitulated in the transcript level. So there is some value then in doing a proteomics study to maybe understand what's really going on at the protein level. Just measuring mRNA about this doesn't really tell you the entire story, and so now we're trying to chase down what all these changes in protein levels really mean as far as biology. Another study, this is now related to our project related to the Department of Energy Institute for Genomics and Proteomics. We're studying this bacterial system. This is a syntrophic microorganism, and in particular this Syntrophicus, which is a system that takes benzoate and butyrate and converts that to hydrogen. So this is another system where we're trying to see if this could be a way to harvest unusual new biofuels. And in our proteomics study then, we found some unusual post-translational modifications that were really unpredicted from just looking at the genome system. This system then, we were able to find these new post-translational modifications based on some recent history of histones. So histones that are related to translation. There had been a study group done by a group at the University of Chicago that had suggested that non-histone proteins could be acetylated by the presence of different CoA esters. And so if you have a CoA ester that has crotonil or acetyl, proteins involved in that are in the presence of these CoA esters could be crotinylated or acetylated that were not predicted in the genome studies. So knowing this then, we had noted that in some of the enzymes that we found when we were doing the general proteomics of this Syntrophicus system, this shows the pathway converting cyclohexane carboxylate from crotonate to cyclohexane. We had noted that we had found many of these enzymes that are present in the system, and there's lots of CoA esters that are present as substrates in the biological pathway. So we were able then to see if any of the proteins that were present in this particular pathway did have these unusual post-translational modifications that were suggested in the non-histone protein studies that the Chicago group was doing. And so what we found was that this just shows a pie chart of proteins that were found to be proteinated. So if we had not known that proteins could be proteinated, we would not have found these proteins. We started off by separating proteins by 2D gels, but then looking at the mass spectrometry data very carefully to see if any potential modifications were present. What we found was 98 proteins were proteinated. 54 of these proteins were related to metabolism. And if you just look at the breakdown of the metabolism proteins, many of these proteins are involved in energy conversion or lipid metabolism. So this study then was the first example then of a prokaryote having a proteonyl lysine modification on proteins. This is the first example of a novel modification. This is a 3-hydroxypyrimidine lysine modification. And there's other modifications that we found that were the first of its kind in a prokaryotic system. Now, what we're trying to now understand is how are all these modifications present in the system? What is the organism doing when it's making these modifications? Is it a way to turn on and turn off certain pathways? This work was entirely done by this one student who actually is trained not at this university, but she's from Ho Chi Minh City, my student Hong, and so she is writing up the manuscripts that describe all of this novel work. Lastly, then, let me just describe some of the basic studies that we did to try to translate proteomics work towards medicine. In medicine, there's a great need then to have a biomarker or a set of biomarkers that can be used clinically to suggest that a patient maybe has a disease or maybe even to guide therapy if the patient for certain diseases. So having a biomarker or a set of biomarkers could be useful for the clinical diagnostic applications for health. At UCLA, we had attempted to use saliva as a way to find markers for particular diseases. In fact, we tried to find markers for cancer just based on measuring one's protein profile from human saliva. For many, many years, we were working with a group of researchers at UCLA, University of Southern California, University of Rochester, University of California, San Francisco, and we had all collaborated together to take human saliva and try to measure all the possible proteins that are present in the salivary, in whole saliva and also in the individual salivary glands. And we published a series of papers. So in essence, there's about between 2,000 and 3,000 proteins present in saliva. These were under normal, healthy conditions. All of the subjects were between 20 and 30 years age. They were all college students, in fact. And we had pulled all the saliva together and just measured all these proteins that were present. We compared them to proteins in plasma. There is some overlap between the proteins in saliva and the proteins in blood, which now suggests that maybe we could use saliva as a way to diagnose diseases not related to the oral cavity as a way to find markers. And so we had started a study. We hadn't really finished it yet, but this was our early attempt to measure proteins present in pancreatic cancer. So we had recruited a series of, we recruited 20 subjects that had pancreatic cancer at various stages, and we found 200 proteins that were unique only to those people that had pancreatic cancer. Now, we haven't finished it yet. There's still a lot of work to be done to try to verify that these are potential proteins or markers for pancreatic cancer. But just the idea to suggest that one could just spit into a cup and tell if a person has pancreatic cancer has some potential as a biomarker for this particular disease. There's a group at UCLA that's trying to develop this kind of a portable device that you put a drop of blood, a drop of saliva on this machine, and you would then be able to tell whether that protein is present as a way to diagnose cancer, for example. So lastly then, in the short time I have, I'll give you one example that we're working on as another kind of biomarker. This is a biomarker for concussion. In the US, it's called traumatic brain injury. And there's many instances of traumatic brain injury that can occur in daily life. So when someone gets hit on the head, that could be a concussion. If you feel a little bit dizzy, you might have a concussion. And if you have lots of these hits, that's not good for your brain function, and sometimes you've got to recover from this kind of a injury. There are no drugs for traumatic brain injury. You can't just take a drug and recover from traumatic brain injury. So there's not a good way to diagnose brain injury. There's not a good way to treat brain injury. In the U.S., there's a lot of people that are interested in having a marker for traumatic brain injury for just common people. So like children playing sports in the field like soccer, athletes, professional athletes that have a brain injury, just trying to diagnose whether that person could even come back and play again immediately is a great use if we had a marker. This is what happens to a brain if they receive repeated brain injury. So this is what happens. You see loss of brain matter. You get this chronic traumatic encephalopathy that occurs, and you can't really diagnose this until the pathology is done. So you have to do an autopsy, unfortunately, to understand whether this kind of injury has resulted. So in the U.S., our military is very interested in having a measurable marker for whether a soldier has received some sort of brain injury. Head injuries right now are not clinically well defined. Right now, the test is a question. They ask you a series of questions to see if your brain is functioning well, but there's no other kind of test right now that's being done. So we would like to have some sort of a surrogate marker, a marker that we can measure in some sort of tissue or fluid or blood sample that would help diagnose whether the patient has received a severe or mild type of head injury. And we're using proteomics then to try to identify what potential markers might be good to diagnose a traumatic brain injury. So the way that we're doing is that we're collaborating with a researcher in neurology at UCLA, Dr. Warner. She is studying astrocytes. So astrocytes are these large cell bodies that are the link between the blood vessels and neurons in the brain. She takes astrocytes from animal models and she can culture them, and from these cultures then she can then subsequently injure them or traumatize them in a way that we can then translate that to the human studies. And so what Dr. Bonner has been doing is that she takes these astrocytes, traumatizes them in a machine. This machine will take the cells, you place them on a membrane, and you put them in a cell. And then you pressurize the cell with some air at a very rapid pulse. When the membrane stretches as a result of the air blast, the cells that adhere to the membrane stretch, and when they stretch, they leak. And so this is a picture of some cells that were on the membrane before you pressurize the cell. And this is what happens to them afterwards. You see that the cell morphology changes, and if I show you more expanded versions of this, you see that the cell membrane leaks, and they form holes in the membranes and proteins leak out. And so the idea is then that once the proteins leak out, if we can identify what these proteins that leak out as a result of this injury, maybe we can find these same proteins in spinal fluid from human subjects or animal subjects, or maybe potentially blood samples. So what we did was we stretched these astrocytes, we injured these astrocytes, and we measured 79 proteins that definitely leaked out of the astrocytes. We compared that to what's known to be present in cerebral spinal fluid from traumatic brain injury subjects. We found 6 proteins that we could classify as potential proteins that we could measure as a potential biomarker for traumatic brain injury. So these are 6 proteins that I can't tell you what they are because we have a patent application pending right now. But these 6 proteins appear to be very specific to severe traumatic brain injury. These are the mass spectrometry measurements of 4 of these markers, A, B, C, and D. And on these graphs, the red bars are from subjects. These are human subjects of traumatic brain injury. And if you look at the levels of these bars in the red, these are all very high compared to these that are down below here. these are supposed to be green. These are healthy subjects that did not have a severe injury in the brain. And most of these subjects that we were able to recruit that were traumatic brain injury subjects have these elevated levels of these potential markers. These are from cerebral spinal fluid. Now, the ideal case would be that we don't measure them from CSF because no one wants to donate CSF samples. People want to measure them in blood. So we've done a few cases now. We can measure a few of these proteins already in the blood samples that we are able to measure. This is one potential marker in a traumatic brain injury human subject from their blood, and we have very good sensitivity for detection of this particular marker. So the idea is then that once we find these 6 markers and verify that they could be potential markers for TBI, we translate that to blood. Now we have a good way now to measure the presence of a traumatic brain injury from these kinds of measurements. By mass spectrometry, we can do this because we don't need an antibody for this protein. We measure a stretch of sequence from the protein directly by mass spectrometry. It's antibody independent. We can measure many hundreds of these potential markers at the same time using mass spectrometry. And so translating then this biomarker platform to the clinic would be then of use then to diagnose, we believe today, severe traumatic brain injury or potentially mild traumatic brain injury. These are the types of injuries that might occur on a playground for children or a soccer field for professional sports. Okay, so lastly then, let me just try to summarize what I've tried to discuss. Proteomics, if you haven't considered it before, offers some unique tools that could be used to address important questions in biology and medicine. Many laboratories specialize in quantitative differential analysis, the measurements of post-translational modifications on proteins, and biomarkers of diseases that could be used in the clinic. At UCLA, we're fairly well equipped, I would say. Now, I didn't say this before, but proteomics is an expensive endeavor. We use expensive mass spectrometers and other analytical equipment that, fortunately, UCLA were well resourced in this particular area. You need instrumentation, you need resources in informatics, and you need the expertise in order to use this equipment. And finally, why I'm here, I'm looking forward to potential collaborations. We're well equipped with the resources, but we're also well equipped to train new students in this area of proteomics. So we welcome any interactions that we have covered while we're here at VNU. These are just some students that are using the equipment at UCLA. Okay, so finally, let me just acknowledge the people that do the work. Our work is funded by various agencies in the United States. The Traumatic Brain Injury Project now is funded by a pharmaceutical company, Abbott Laboratories, that's trying to use our markers to develop a handheld device for traumatic brain injury. And this is my research lab. This is Hong, who was doing the work on post-translational modification. So I thank you for inviting me, and I certainly help welcome any questions that you might have about the work.